Good morning, everyone, or good afternoon if you happen to be in Switzerland or Europe. We're so excited to welcome everyone here and to highlight some of these amazing startups that are coming through various Swissnex programs. My name is Alicia Evangelista. I'm the head of innovation here at Swissnex in Boston, and I'm going to be your host and moderator for the day. We have a number of startups to hear from today. We're excited to be able to welcome both startups that are in Switzerland at the moment physically and those that are also here at Swissnex in Boston over the next week and potentially through the week after. For those of you who are in the audience, we will be launching a poll after each startup has their opportunity to present. If you are interested in reaching out to them or in hearing more from them, please answer yes to the poll and we'll make sure that your information gets to them. We'll also be accepting questions from the audience for a Q&A session following each presentation. Feel free to drop any questions into the Q&A as they come to your mind, and I'll make sure that they get read to the startups and they get addressed as best as possible. So today we are featuring two different programs that are coming through Swissnex. The first is a cohort of startups that are located in Switzerland at the moment, and they're part of the Innosuisse program, which is a government-funded startup support program that is meant to help startups both establish themselves in Switzerland and come to the US. And so many of these participants will be traveling to the Boston or in some cases San Francisco area later in the year or next year, and we're happy to connect you. We'll also be hearing later on from the Venture Leaders cohort, and I'll allow Jordi to, their program director, to introduce himself in a little bit. The Venture Leaders are here in person, so we're looking forward to hearing from all of them. But without further ado, I would like to get the program started. Our first presenter is going to be coming to us from Switzerland, and I'm just finding him. Tino is the CEO of Anavo Medical. Anavo is creating a new way to treat seromas that occur in breast cancers or in mastectomies. So Tino, if you can hear me, the floor is yours and you're welcome to share your screen.
Thank you very much. I cannot really start my video at the moment.
Can you share your screen?
The host, yes, I can try. Yes, can you see that?
Yes, we can.
Awesome. I hope the internet is going to work fine because we're in Lisbon at the moment, so at the conference, so it might be a bit patchy, but yeah. All right, nice to meet you, everybody. I'm Tino Matter from Anavo Medical. We are tackling seroma formation, an issue that affects millions of patients worldwide every year. One of these patients is Victoria. Victoria was diagnosed with breast cancer at age 36. Surgeons took out part of her breast to fight the cancer, and she was able to recover at home. 1 week after the surgery, though, she felt pressure under her sutures, and liquid started to explode through her stitches. Alarmed and shocked, she went to the hospital, where they diagnosed her with seroma, a chronic accumulation of liquid right under the skin that doesn't resorb. To get it treated, she had to go back to the hospital over and over again after all the emotional and physical stress she had to handle. Or, as she puts it herself, the last thing you want to do is to go back to the hospital for yet another needle. It sounds like a horror story, but Victoria is not alone. There's more than 2.5 million procedures every year with a very high seroma risk in the EU and the US alone. What Victoria has entered is the so-called vicious cycle of repeated seroma aspiration. If a seroma forms after surgery, as you can see in step 1, a surgeon usually aspirates it with a thick needle and hopes that it doesn't come back. More often than not, though, it does come back. Along the way, you're faced with a delayed cancer therapy, a fibrosis risk, and an infection risk. And the surgeon has to aspirate it over and over again. Along the way, you have all these risks. In a groundbreaking collaboration between leading research institutes in Switzerland and surgical leaders in the field, we have developed a nanoparticle-powered solution to solve this issue by injecting particles directly into the seroma after the first aspiration and therefore healing it helping the patient to recover faster, doing so cheaper than a standard of care, and overall offering a higher quality of life and satisfaction to the patient. We have shown in a rat model that there's no toxicity, that the particles don't clear to the organs, that there's no systemic effect. So the effects of the particles are strictly limited to the seroma cavity. Additionally, we have shown that the particles heal seromas after 1 week, as opposed to the standard of care with a 35% rate and fibrin glue with 50% rate. Our particle heals seromas within 1 week with a 100% rate. And we also looked after 6 weeks at the incision. And as you can see, the wound healing is so strong with our particles that you can barely see that there has been any surgery performed on the rats. Who is we? You heard myself talking quite a lot. I've been always passionate about innovating materials to improve people's life. I've done my PhD on this topic. I'm the co-inventor of the technology. With me on the team is Sebastian Loy, who has a strong business background, and we're completed by researchers, clinical partners, and scientific advisors. We have a clear plan going forward, all the way to a product launch in 2026. And with this, I would like to close you just a small call for action. We're closing a 3 million seed round next year to establish GMP production, to run large animal trials, and get ready for clinical trials. Thank you very much.
Thank you, Tino. As the audience thinks about what questions they might want to ask, I think we can jump in, and I'd like you to talk a little bit about the regulatory status of your solution right now. Where are you within the process, and what help might you need to get through a designation?
Yes, sure. So we are currently in the preclinical phase. So we're doing animal studies for both toxicity and efficacy. We have a large animal trial in sheep planned early next year. And by the end of next year, we plan a, I'm just going to click away this poll. we are going to do GLP studies, so the confirmatory studies before going to clinical trials. We expect clinical trials to start in 2023 as well, and to complete the first one by 2024.
Fantastic. And in terms of the Boston ecosystem, what sort of partners or support are you looking for?
Yeah, currently we have quite a strong clinical network in Europe. We are looking for surgeons that are interested in this topic in the Boston area as well, that can give us input onto the project and the product itself, to see where their biggest pain is and how the product has to be designed in order to maximize the effect. We're also interested in understanding the exact process within the hospital with these patients. So if you have somebody a bit more from the operations perspective, that would be great. And obviously, since you're closing a seed round mid next year, also funding would be interesting for us.
Great, thank you so much. I don't see any other questions, so I think we can move on to our next presenter. But thank you, Tino, for sharing, and best of luck at the conference.
Awesome, thank you very much.
Thank you. So our next panelist or presenter will be Benedict Vonner with Simplechem. Simplechem is a little bit of a change in direction. It's a research tool. And we'll be hearing from Benedict about how they're hoping to launch in the US, and hopefully some exciting news about their recent partnerships. Benedict, are you here?
Yes, thanks. Also cannot start my camera, but that's fine. Tino made it like this, so I also will be happy to do like that. Yeah, hi, my name is Benedict from Simplechem, and we have basically created the most easy and efficient automated chemistry system for the discovery of new drugs and materials. When new drugs or medicines are invented, they go through several stages, and today most of these stages use a lot of automated methods. And just here in the middle, in the lead optimization, especially in the make part, there is still an army of chemists who manually and very repetitively synthesize many different variants of new molecules. And this basically creates a bottleneck. Efforts to automate some of the synthesis work have been made, but the solutions are all really complex for actual chemists to use. So I would like to make an analogy between chemistry and coffee making. So existing solutions can be actually represented by really complex barista coffee machines. They are too big, too expensive, and really way too complex for the requirements of a normal user. And essentially, there are a solution for specialists. So for the user, these, like for a user like me or for a chemist, for example, capsule coffee systems are great. They're affordable, small, and easy to use. And we basically apply this concept to chemistry. In our simple system, to make a new molecule, the user simply inserts the starting material he would like to transform, then a disposable single-use reagent cartridge that contains all the reagents for the desired reaction. And then upon the push of one button, the machine carries out the chemical reaction to generate really the product in a short time, safe and convenient manner. And this enables researchers with even little chemistry expertise to rapidly synthesize molecules, enabling faster and more efficient development cycles. With this, we could already convince customers mostly in the pharmaceutical industry and contract research organizations, mostly in Europe, and we have about 30 instruments installed so far. How did we get there? Just a rough overview. Simple was founded in 2016, and we realized a financing round in 2017 to run a pilot phase with our Simple 1 instrument, and then developed our commercial Simple 2 in the end of 2019. And since just very recently, our products are now also available worldwide through Merck or Sigma Aldrich to make it very easy for the customers to get their hands on the technology. And also get in touch with us if you want to find out more what's next. And we are working basically on a new AI-driven applications for our technology that will make the technology really even easier to use. Over time, we are also able to assemble a strong team where Paul is in customer relations. We recently were joined by Christian and Thomas, who are on the operative side. Samuele, Guillaume, and Anna, together with two, are constantly developing new types of cartridges, new types of applications for our users. So I don't want to take too much of your time. We have started recently to bring the system as well to chemists in the US. So if you know anyone who could profit from the system, we'd be very happy for an introduction. And also for our AI-driven applications, we are looking for some funding or potential partners who would like to work with us. So don't hesitate to contact us for more information. Thanks very much.
Thank you, Benedict. And I think you should be able to turn on your camera now. We fixed our setting. I think the first question is, so I know that you're coming to the US in early next year, I believe. Is there a time frame when people might be able to meet you in person?
We're still waiting for that offices open up, that people can, that we can visit them at their company site. But yeah, if anyone says, hey, please come over before, we could meet not in the office, but somewhere in the coffee shop, then we can start making plans that we can move ahead faster.
And great. And what's the learning curve for learning how to use the device? Is it really as simple as using a Keurig or a Nescafe, or is there a significant amount of training?
It's as easy as that. We have initially, for instrument, we have traveled to the customer to deploy the system. But now all we need to do is a 10 minutes video call to help the customer to set it up, and then they can run it by themselves.
Great. And I know you had a piece of news with a nice partnership recently. Do you want to share that with the audience?
Yeah, so yeah, Merck is now basically distributing all our products, so they can be ordered through their homepage, and that's very, very good for the customer, since they don't have to go through the process of adding us as a supplier, but they can just put it in their take a credit card out, order the instrument, some cartridges, and you can start a couple of days later.
Great. All right. Well, thank you so much, Benedict, for walking us through Simplechem. I hope that some people in the audience will consider making their lives a little bit easier if they're doing medicinal chemistry, and will get in touch.
Sure. Thanks.
Thank you. Our next speaker is also joining us from Switzerland. We have Dr. Utkud Gulan, who is with High-D Imaging. And High-D is a platform that's looking at medical imaging processes to improve surgical planning and outcomes in the cardiovascular system. Utkud, are you here?
Yes, thank you, Alicia.
Great. And I think you should be able to share your video and
I can't because it says that other participant is sharing.
try again.
Yes, perfect. Can you see my screen?
Yes.
Very well. Thank you very much. Right now, billions of hearts are beating all around the world, and at the same time, under different conditions. And each one of us has our own unique heartbeat. And what do we all want at the end of the day? To be healthy and active so that we can play catch with our grandchildren. However, this is not possible for so many of us. Why? Because globally, more than 10 million patients suffer from a severe cardiac disease called heart valve dysfunction. Every year, 500,000 patients undergo heart valve replacement operations, and 1/3 do not show improvements, meaning they live with severe lifestyle disruptions. 1/3. It's a huge pain, not only for patients, but also for physicians. Today, doctors still lack data to better understand the true conditions of a patient's heart before they operate. And there's a strong need to improve the efficiency of cardiac planning. Hi, I'm Dr. Utku Gülen, CEO and co-founder. This is why ID Imaging was founded. We help doctors create fast, accurate, and personalized cardiac planning within seconds, thanks to our advanced AI software. Think about it. By seeing a patient's heart even before they operate, doctors can not only plan and predict their patient's outcome, but they can also dramatically reduce complications that patients experience after surgery. In 2021, we introduced our novel technology to 9 hospitals with 3 paying customers across Europe and the US. And this is just the beginning. We plan to rapidly expand and reach over 50 hospitals globally next year and focus on the US market. Our vision is to provide healthcare professionals our decision-making software for the diagnosis and treatment of all cardiac diseases, which has a global market of $25 billion. And within 5 years, we plan to penetrate 10% of our potential market. We offer a highly scalable business model based on SaaS to all our current and potential stakeholders. In order to build such an advanced technology, you need a unique team. I'm honored to lead a fantastic team that accumulates over 50 years of experience in the fields of business, engineering, and cardiovascular diseases. We at ID Imaging are determined to be the first to bring tomorrow's AI-based technology today to hospitals all around the world. Join us, our physicians and partners, to save lives and make sure that every grandparent can play catch with their grandchild. Thank you.
Thank you, Utku. I think to start, can you tell us a little bit about your journey in the US? Because I know you've already started to work on having a presence here and are part of a few programs.
Absolutely. We are part of 4 different programs in the US. And soon, hopefully next month, we will start our first clinical study with a patient size of 200 patients in the US. And we also would like to bring our product to the market within summer next year.
Fantastic. And for the people listening in on this call, what would be the most helpful connection for you to make today?
We are looking for partners, particularly investors for our seed round, and also strategic partners to market entry, and finally, stakeholders from hospitals and key opinion leaders who are experts in the cardiac field.
Great. Is there anything else that you'd like to share with the audience? I'm seeing that people are very excited about your solution, and I am as well, but anything you want to add for them?
Thank you very much. We look forward to connecting you. Please contact us, and we will be happy to join you our journey.
Fantastic. Thank you so much, Utku. We'll be joining to the stage next, or bringing to the stage next, I should say, Patrick Kessel of Perivision. Perivision is a startup that's looking to improve glaucoma diagnostics through the combination of AI and VR. Patrick, are you here? Here he is. Patrick, the floor is yours whenever you're ready. Patrick, can you hear us?
Yeah, I'm
Great.
just trying to share my screen here. So, this is not working. You cannot see my screen, right?
No, we cannot.
So, here we go. Fantastic. Hello, everyone. I'm Patrick Kessel, co-founder and CEO of Perivision. We're a Swiss medical technology startup that wants to reimagine glaucoma monitoring and deliver it in a much more patient-friendly and cost-efficient manner. Glaucoma is a chronic eye disease that leads to irreversible blindness. Shown on the picture on the right, you slowly lose your peripheral vision, and with it, the window to the world around you. It's a terrible condition for individuals, but also a very costly one for healthcare systems due to its prevalence and the chronic nature. For instance, in the US, it's responsible for more than 10 million hospital visits per year and 6 billion in treatment and prevention costs. So the typical glaucoma patient must check their visual field every year to assess how much they see. But the visual field testing or perimetry process is a major bottleneck. It's long, which reduces the capacity in clinics and practices. The data quality strongly decreases with test time, and it requires an assistant, a dedicated room, which are costly. So if you shorten the exam and provide more testing opportunities, you can make a real clinical, but also commercial impact. At Perivision, we really want to deliver this step change in perimetry for patients and doctors. We perform the same procedure, but in 60% less time based on our AI algorithm. We do this on a light and patient-friendly headset. Through this portability and also the speed, we can offer more frequent testing, for instance, also at home. And with this, we reduce the data variability. And we do not require a dedicated room or an assistant. This also aligns well with trends towards remote patient monitoring. The key opinion leaders in glaucoma want faster and more frequent testing to better understand the disease progression of their patients. At the same time, patients are already performing, for instance, eye pressure measurements at home. So why should they also perform an easy eye test? We want to first optimize the current workflows and practices in clinics, and then we want to build entirely new care pathways through new points of care, for instance, at home or also in opticians stores. Now, the current market for these monitoring devices is roughly 430 million big, but with our solution, we can really scale up and expand this market to 680 million by solving the capacity constraints and also offering these new care pathways. Now, so far, we have patented our AI testing algorithm and validated our technology in 2 clinical studies. We have also secured letters of intent from various private practices here in Switzerland, and we are building a customer pipeline to visit in the US next February. The next 2 years, we have to finalize our product and obtain the regulatory approval for CE Class 2A and the FDA 510K exempt. And at the moment, we're really looking for further introductions to US eye doctors, optometrists, and also investors to raise our seed round. Now, to execute our strategy, we have also all the necessary clinical, technical, and business expertise. We're supported by a world-class expert team in AI, perimetry, and ophthalmology. We're Perivision, and with your support, we believe we can make a huge difference for glaucoma patients and doctors worldwide. Thank you very much.
Thank you, Patrick. let's start with a question about what role that AI is playing in the solution. So it's a fairly unique way to approach glaucoma.
Right. I can also pass this question on to Sharifa, who is also here, our co-founder. She is the CTO and has developed the AI algorithm.
We're just bringing her up as a panelist. Give her 1 second.
Ah, sorry, that doesn't, okay.
That's okay, she's here. Sharifa, would you like to answer the question of how does AI play a role in the solution?
Yeah, thank you very much for the opportunity. Yes, AI actually makes the test like really faster than it is. It actually reduces the examination time by up to 70%, and it also, as we can reduce the examination time, it also delivers high quality examination for the patients, because these patients are generally very old people and they can be affected by this long examination, and this reduces the performance of the test highly. So it really affects first, of course, the comfort of the patient, and then of course the quality of examination.
So you touched on this a little bit. So just to reiterate the value proposition, it's time, it's comfort. What else?
It's also, of course, it also affects the cost, because it reduces the examination time. So you have less time for the assistants, and then it can save time and cost for the clinicians and assistants time, which can allow to increase the capacity of the clinics.
Great. And are there any other products on the market that are using AI for glaucoma? Is there anything even remotely close?
Not really. This is actually our unique selling point.
Great. All right, wonderful. Thank you so much, both Patrick and Sharifa, for joining us.
Thank you very much.
Yeah, it's a pleasure to have you. So we're going to move on to a completely different application. So our next company is Terrapet, and we're going to be joined by CTO Marcus Palm. Terrapet is a spin-out from CERN in Switzerland, and they're developing a novel medical device for proton therapy. So Marcus, if you can hear me, the floor is yours. You're on mute. Marcus, you're still on mute.
There we go. Sorry. Can you hear me all right?
Yes, we can.
Okay, perfect. So hi, everyone. Sorry for that. My name is Marcus, and I'm co-founder and CTO of Terrapet. And at Terrapet, we are developing a novel medical device that will act as the eyes for the doctors doing proton therapy for cancer treatment. Now, proton therapy, this is a new technology using high-energy protons to treat cancer. And it's the most precise form of radiotherapy that can provide patients with minimum damage to healthy tissue and organs. But although proton therapy itself can be extremely precise, the treatments today actually only rely on simulation, and there is no real-time monitoring during the treatment. And in practice, this means that the doctors are not 100% sure about where inside the patients the protons actually arrive. And because of that, the doctors, they have to be conservative when they plan the treatment. They have to add large margins around the tumor just to make sure that they really kill all the cancer cells. But of course, this means also unnecessary radiation to the patients. So essentially, if the tumor has the size of a clementine, why would you treat it as an orange? And this is where Terrapet comes into the picture. So our product is a sort of add-on medical device to proton therapy facilities. So this device will tell the doctors where inside the patient the protons arrive in 3D, in real time, in vivo, and in a non-invasive way. Because when you know where the protons actually stop inside the body, you can use this information to reduce these margins around the tumor, which will mean safer and more precise treatment for the patient. But our story doesn't really stop there, because our core technology is really a platform technology, and that has applications beyond proton therapy. For example, like preclinical and clinical pet markets, which are even bigger than the proton therapy market itself. So we believe that we have a very good potential to keep growing beyond proton therapy in the future. The team consists of, we have experienced physicists from CERN. We have renowned field experts from proton therapy and radiotherapy, and we also have businessmen on board. And we are strongly supported by the Swiss startup ecosystem as well as on a European level. And we are confident that we can bring a company to the next level. We're also growing very fast. Within less than 1 year since the incorporation of the company, we managed to raise more than 3 million. And today we're about 10 people working full-time on the development of the first full-scale prototype for CE certification in Europe. And we're also collaborating with, for example, with CERN, Physics Institute, and the Nobel Prize awarding Karolinska Institute. And right now we are raising our Series A round. More than half of the round is already completed. So I would like to quit with taking this benefit to invite new investors to. Thank you very much.
Thank you very much. When is the last time that there was a significant change to proton therapy, or is this the first of its kind in quite a long time?
I think the biggest change was actually started in Switzerland at the PSI Institute. They started doing something called spot scanning, which means that you basically, it's a bit like 3D printing, or you divide the target into three-dimensional spots, and you target the tumor spot by spot, which makes it possible to conform the dose much better to the tumor compared to how it did before. But that's already more than 20 years ago that started doing this at PSI. And about 10 years ago since it became more widely available.
Great, thank you. How do you combine proton therapy with PET imaging?
Yeah, that slide was more related to different applications of our technology. That the technology that we use has one field of application with proton therapy, but it's also useful for like conventional PET scanning technology.
Great, thank you. And so you mentioned that you're looking for investment. What else are you looking for from the US market?
Yeah, we would of course be really interested in getting a foot into the US market as well. It's like one third of the world market is in the US for proton therapy, and it's also growing quite fast now in the US. There are new facilities popping up. So yeah, of course, we would be very keen going into the US as well.
All right, fantastic. Thank you so much. I think that there will be some interest from the audience there as well.
Thank you very much.
All right, we're going to take a moment now to switch locations. So we have heard from all of the startups that are in Switzerland at this point, and now we're going to start hearing from the startups that are in Boston. So I'm welcoming to the stage Jordi Montserrat, who's the Managing Director of Venture Lab, and he's going to introduce their part of the program and the history of Venture Lab, which is, I think, 16 years rich in Boston now. Jordi, it's all yours.
Yes, hello, Alicia, and thank you very much for welcoming us. It's a pleasure, effectively, to be back in Boston. We were here a little bit more than a month ago, and we had the chance to come back now with a biotech team. So it's my pleasure here to make a quick introduction. And therefore, just allow me to start my presentation, I think, with these few words. Many of the people in the audience are probably familiar with the venture leaders and actually what we do at Venture Lab. Let me just share rapidly, for the past 15 years, we have been building platforms, tools in order to help the most promising entrepreneurs that have global ambitions to reach their objective, particularly when it comes to go international, to raise the necessary financing, and to meet partners in order to do this. Just to mention Beyond Venture Leaders, which is the platform we're here with today in Boston, we have built as well, and we have the chance to run the Innosuisse startup trainings to have the company starting module 3 and module 4. Many of the companies that you have seen just in the prior sessions are also part, of course, of these through Innosuisse, supported by Innosuisse. We have the Venture Kick platform where we do pre-seed investments, over 90 projects per year. We invest more than 5 million. It's been going on for 15 years now, and a very promising pipeline of companies who are here, and we do the early stage financing. And finally, and this will be of most interest for many of you, reach as well to the top 100 Swiss startup. Every year we publish the ranking of these very promising companies across the board, not only biotech, but this is something that brings to the international investors the possibility to have a deep dive into the Swiss startup ecosystem. And today, to mention, it is published not only in English, of course, German and French, but also in Chinese and Japanese, and therefore really the possibility to dive into this ecosystem. With this, we are here with the Venture Leaders, and it's my great pleasure to introduce the team, Venture Leaders team of this year, Venture Leaders Biotech Team. 15 years we come here in order to foster this bridge between Switzerland and the US, particularly Boston, of course, when it comes to the life science sector. And it is my pleasure to give the floor then next to Avalogix, Actera, Alitea, Abrion, Nemozia, Nex Immune, PBNB. And we have a small error with the thing, so it will be another 8 company which are here. But I mean, as said, coming here, making the link with Boston, and having here the possibility to bring them. They are now in Boston. For those who are in Boston right now as well, the opportunity to reach out to them and to send them contacts. I mean, contact them. You have the possibility to meet them all of the week with this one. So with this, and actually here, yes, so the company it's CellVA we're missing. Sorry for that. We're missing the logo for CellVA that will be presenting as well today. Now, all of this, I would like to conclude this introduction by, of course, thanking the partners, Swissnex, of course, to mention we're co-organizing with this week here, but also all of the supporters who are sponsoring this edition, who are making it possible, in particular, of course, EPF LETH, the canton of Zurich, Vischer attorney firm in Switzerland, and Morrison & Foster attorney firm here in Boston and in the US as well. If you want more information, don't hesitate to reach out to Venture Leaders. You have the contact, the website right here. We're happy to connect, and we will be back next year, as we have done all the past year, continue fostering this bridge. Our history shows today we have many investors who have invested in these companies, so use this track record to build the next generation of success stories together. Thank you very much to Swissnex Boston, and Alicia, I'll pass you back the word. I will stop here this introduction. Thank you, Alicia.
Thank you so much, Jordi. Appreciate the intro as always. And we're hoping so much next year that we're able to do this in person and get back on that yacht and have a good time with everyone. But that having been said, the next presenter is from Avalogix. We're welcoming Ignacio to the stage. Avalogix is developing monoclonal antibodies against novel biological targets. Ignacio, the floor is yours.
Can you see my presentation?
Yes, and we can hear you now. Go ahead.
Okay, fine. Okay, thank you very much for attending our presentations today. I'm going to introduce to you our company, Avalogix, the spin-off oncology company from the University of Geneva. We have several products in development, all at the preclinical level. But today I wanted to focus on our lead asset, monoclonal antibody H225 for the treatment of lymphomas. It blocks a target called JAMC, junction addition molecule C, which is a biological target that sits in the tight junction of the endothelium. 1 second. Basically, the medical problem we're trying to address is the problem of non-Hodgkin lymphoma, which is the 7th most common cancer in the US, with a market potential to reach about US $15 billion by 2026. Now, there are medications in the market. Rituximab, for example, is one of them. BTK, Proton Therapy kinase inhibitors also are in the market to treat the non-Hodgkin lymphomas. But the reality is that 80% of patients become resistant to established therapies, whether it's rituximab or also BTK inhibitors. So there is a captive market that's potentially very, very interesting for those patients who become resistant to these therapies. Now, what we did is we looked at this protein, JAMC, junction addition molecule C, and we look at its overexpression in certain malignant B-cell cancers. We took 250 patients, and these are acronyms for some of these different lymphomas. And we see that in some lymphomas, this protein is highly expressed. For example, mantle cell lymphoma, 75% of the patients overexpress the protein, MCBL, and then also some others. So we've decided to focus on mantle cell lymphoma. Why? Because the unmet medical need is huge. These patients die after they fail on a first-line treatment. Also MCBL, because about 80% of the patients are JAMC positive. And then CLL, where maybe only 15% of the patients are JAMC positive, but nonetheless, it's a very, very important indication. Basically, this is the one scientific experiment I wanted to show you. It's a typical mice model, where basically at time 0, we engrafted mice with Cheka1 cells, which are mantle cell lymphoma cells, and we started treatment at the day 5 with either an antibody or a control antibody. And we can see that when we treat with a control antibody, the animals die after 40 days. And with our antibody, they also eventually die, but we can see that at day 62, we still have a substantial number of animals that are still alive. This gave us hope that we were on to something interesting, and that's why we decided to create the company. We've looked at the market potential of the product, given the competition. Obviously, the US is the highest potential market. We estimate that with a product launch in 2027, we can reach a peak year sales of over $1 billion in these 3 different indications. And the first one we're pursuing, as I mentioned before, is mantle cell lymphoma. This is our development plan. These are our capital requirements. We require about 20 million to get to market. We plan to do 1 single clinical trial in the case of mantle cell lymphoma with about 100 patients to get a product approval. And this is being done in oncology these days, where for very targeted products like ours, 1 single clinical trial, 180, 120 patients is accepted by FDA to get a product approval. Our team and myself, I have several years of experience in pharma and biotech. Professor Imhoff is the scientist behind all of this, and he's our chief scientific officer. And then we have Jan Dien, who's a senior project development leader with many years of experience in the development of biologicals. Future additions in the years to come will be an expert in the manufacturing of biologicals. Manufacturing is a big issue with antibodies, a chief medical officer, and then we also plan to subcontract other functions such as IP, regulatory affairs, and so on. So thank you very much, and I'll be glad to take your questions now.
Thank you, Ignacio. I think that the first question is, can you talk a little bit more about the mechanism of action? Is the antibody actually affecting the tumor size, or is it working in another way?
Right, the antibody basically it's blocking this protein called JAMC, and basically the cancer cells have developed the ability to express this protein in their surface in order to migrate through the blood cells into the target organ. So basically what we're doing is we're blocking metastasis here. We're basically blocking the ability of the lymphoma cancer cells to home in the target organs, which are spleen, lymph nodes, liver, and a few others.
Great. And so it sounds like there's opportunity there for combining this with existing treatments. Is that the plan?
Right, that would be the plan. I think that obviously when you develop a new therapeutic, you hope that it will work by itself, but the reality is that the way things are going now, we're seeing more and more combination therapies. We have checked in vivo models and synergies between our compound and checkpoint inhibitors. And there seem to be some synergies, and we'll be exploring more of that in the future.
Fantastic. Thank you, Ignacio.
Okay.
We'll be welcoming to the stage next Pierre-Alain from Acthera. Acthera is creating novel hard-shelled liposomes for targeted delivery of therapeutics. Do we have Pierre? Ah, there you are. The floor is yours.
Hello, everyone. Let me share my screen. Voila. So, thank you, everyone. Do you see the full screen or the presenter screen? Thank you. Okay, now it should be right. So thank you, everyone, for giving us some time. Oh, that's strange. Well, we can continue like that. Let's be honest. Voila. I don't understand why.
I think we can see it well enough if you want to.
Well,
continue.
yes, well, yes, let's continue like that. Sorry for that. So basically, thank you for your time. And my name is Pierre-Alain Monard. I'm a co-CEO, CSO, and co-founder of Acthera Therapeutic. And basically what we are intending is tackling one of the basic challenge that doctors are facing every day in the treatment of patients. The fact that if you deliver or if you administer a drug by IV in a systemic fashion, only a very small portion of the drug will really have a therapeutic effect. So this leads actually to very high dosage that needs to be basically processed by the body. And this is a clear burden on the body, but it's also leading to very high numbers of side effects. So imagine for 1 second, if instead of having 2% of the drug reaching the intended target, we had 20%, 40% of the drug, which means that the doctor wouldn't have to make a choice. Do I administer the best drug, but the one with the most side effect potential? Or can I use the right drug? For the patient, imagine that, being able to be treated, knowing that you have the best chances to basically win your fight against illnesses. So this idea of targeted delivery is an idea that we follow, proposing a new vision to this old tool, the liposome. We have basically liposome that will release the payload, the drug, on command. It means that if they are exposed to various external stresses, physical stresses, with a correct energy level, we can decide where and when we deliver the doses. And here we have various ways actually to have this trigger being applied. It could be linked to a pathology, or it could be an external source of energy like ultrasound or light. So the doctor now would be in charge of defining where when he wants to treat the patient. We have already shown in pilot study, efficacy studies in animal, that the concept is working. So where are we? Well, we are in a phase fully funded until the mid of next year, where we are gathering evidence about the concept, demonstrating it in cardiovascular oncology and RNA modality. Middle of next year, we will have to end a financing round that will allow us to carry all the intended study to bring us in clinic by 2024. We have already assembled a team that has all the expertise needed to go there, but we have also the chance to be supported by very knowledgeable medical doctor like Ruggero Della Vita, a former oncology of Roche and Genentech, or people that are specialized in the manufacturing of those systems, the IP, or even the regulatory pathway. So apparently you didn't see the slide, and I'm very sorry about that. So I hope my voice was enough.
Thank you, Pierre. I think, yeah, we didn't get to see the slides, but the story you told is compelling. Maybe a quick question about the triggering of the liposomes. Can it be triggered by pathological conditions, or do you need an external stimulus?
So basically, obviously, the number of pathology where the triggering is, so to speak, pathologic is very limited. But indeed, if you have, for example, a stenosis, so basically a problem of constriction of an artery, this, depending on the stage of the stenosis, will be enough to trigger the release of, for example, an anti-clotting agent, anti-febrile agent, thrombolytic, as it's called, on the spot of the stenosis. And this we have been able to show in animal already.
Fantastic. And have you been able to show the other modalities of triggering? You mentioned light, I believe.
Light has been shown in animals too. In the brain of mice, we have shown that we are capable of basically triggering the release, in this case of dye. So it's more, it's less an efficacy, it's more a proof of mechanism. For the ultrasound, the study are being carried out as we speak.
Fantastic. And what are you looking for from the US? Are you considering other indications, or are you going all in on the cardiovascular partnerships?
So clearly at this point, we are really trying to demonstrate the feasibility of this concept in several indications, as I said. So oncology and RNA therapy, or RNA modalities as they are called. What do we expect from the US is clearly we are looking for partners. I mean, we are not a drug developing company. We are basically a drug system developing company. So we have the HSL, we have the triggering system, and we are open to test actually molecule that as a company would have. That's one point. The second point is obviously we are interested in setting foot in the American market. So clearly having on the one hand investor, on the other hand contact with some big hospital system to carry out clinical study would be fantastic. So we are open for, in all those three cases to discuss with other companies and other entities.
Fantastic. Thank you so much. And best of luck with the first indication. We're looking forward to hearing more. So
Thank
we'll
you.
welcome.
Bye-bye.
Thank you. We'll welcome to the stage now Ricardo from Alatea, which is an organization that is changing gears a little bit. So we've heard from some pure biotech companies, and now we're going to hear from a company that is providing support to biotechs and other researchers. So the floor is yours, Ricardo.
Thank you very much. Can you hear me?
Yes, we can.
Yeah, perfect. All right. So hi, everyone. My name is Ricardo. I'm CEO and co-founder at Alatea Genomics. At Alatea Genomics, we are actually developing technologies to enable the generation of big RNA data for biomarker and drug discovery. So why do we like, first of all, why do we like RNA? RNA is a great molecule because it carries both types of information, both genetic and functional information. As such, and also because it's quite an explored field, studying RNA molecules and generating RNA data provides a lot of potential for the generation of new biomarkers and new drugs based on the study of these molecules. The problem and the challenges with RNA data is that in order to develop new biomarkers and new drugs based on RNA, you normally need to process a large number of samples, for example, from large biobanks or drug libraries. The challenge and the problem now is that the products that are available in the market are not at all well suited or designed for processing a large number of samples. On one hand, you have the legacy solutions such as microarrays, which is great because it's high throughput and low cost, but it's very biased because they're designed to measure only a sub-portion of all the RNA molecules that you can have in a sample. On the other end of the spectrum, you have RNA sequencing, state-of-the-art technology, completely unbiased. You can measure all the RNA molecules in your sample, but it comes at a high price tag, and it's very laborious. This is what we actually addressed, this trade-off. This is what we addressed with our technology called Brb Sequencing, which brings for the first time an unbiased measurement of the RNA molecules in your sample with, at the same time, high throughput and low cost operations. We are already actually on the market. We have 2 kits in the market that allow you to prepare and generate RNA data from tissue RNA and blood RNA. And we're actually working on 3 new products that we're going to put on the market by next year, which is going to enable the generation of RNA data on different types of products, cell lysates for very high throughput drug screening from pharma companies, FFPE samples from patient biobanks, and even single cells. The common theme for all our products is that they really enable the generation of this big RNA data that we need for new biomarkers and new drugs. Just to give you a snapshot of the main application areas and where we are with the company now, we are targeting mainly now the markets for biomarker discovery and drug screening. And we also see potentially actually in the long term to use our technology for diagnostics. We have already, as I was mentioning, 3 products on the market. Since incorporation in May 2020, we have generated a total of 200,000 francs or dollars, and we have 20 customers globally, including top 10 pharma companies such as Johnson & Johnson, Novartis, and AstraZeneca. We are now looking for a 1.5 million francs seed financing round to consolidate our P portfolio, take the new products on the market, and scale up marketing and operations. With this, I'll be happy to take any of your questions, and I thank you for your attention.
Thank you, Ricardo. We had a question yesterday when we were discussing this about your business model. So if organizations are interested in taking advantage of your kits, are they able to run them in-house? Do they work with a CRO or do they work with you directly?
This is really up to them. What we see from our customers that depending on their level of expertise, they actually normally either decide to import the technology in their facility. So in this case, they just buy our kits, buy the kits from us, or they normally rely on service providers. And in this case, it's very easy for us to get in touch with the service provider, establish our kits or our technology at the service provider, and then the client can easily basically send their samples to the service provider, and then they can generate the data for them.
Can you give us any preview of what the next products might be that you would use the funding to leverage or to build?
Yeah, so as I was mentioning in on our slides, basically the products that we are developing are aimed at opening new scenarios, a new type of samples that can actually enter the workflow. And these samples are mainly cell lysates, so people that actually have a lot of samples do not need to go through the trouble of extracting the RNA before inputting the RNA in our pipeline. This is really for big screens and for pharma companies. And also the other product is FFP samples. These are patient samples that are very challenging to work with because the RNA that comes out of these samples is heavily degraded. So we require a little bit of a special workflow. And these are the products that we plan to put on the market by next year.
Really fantastic. I love this product, so I'm very happy to hear you present it again, and thank you for sharing with us.
Thank you.
Great. So we'll be welcoming to the stage next Avalogix Therapeutics and Maximilian. Avalogix is developing precision gene therapy solutions to address genetically defined neurological disorders. Maximilian, if you can hear us, the floor is yours.
Yeah, I can hear you. Can you see me?
We can see you. Not your presentation yet, though.
Yeah, let me share screen. Can you see my deck?
I think, yep, now we can.
Okay, so good morning, everybody. I'm Maximilian Moroni, CEO and co-founder at Proton Therapeutics. We are a spin-off from the Swiss Federal Institute of Technology based on more than a decade of research at the Brain Mind Institute. So our focus is adeno-associated virus-based precision gene therapy to address neurological disorders. So what makes us unique is a novel gene therapy vector that will express the therapeutic transgene in selected central nervous system cell types, neurons and astrocytes in the first version, to maximize, on one hand, the therapeutic efficacy of the treatment, while at the same time reducing risks of unwanted side effects in non-targeted tissues. And there's a number of neurodegenerative disorders which could benefit from this co-targeting of neurons and astrocytes. Now, to lead this venture, we put together a team with expertise in drug development. And you have here the logos from the management and board members where they've worked in the past. What is important is that we have on board expertise in AAV-based gene therapy products from bench to market, with the former CSO of uniQure as Chief Development Officer, and he brought Libera to the market. So with this in mind, the first neurodegenerative disorder we decide to address is a common rare disease called amyotrophic lateral sclerosis. The British astrophysicist Stephen Hawkins developed a rare form of ALS. Basically, it's a deadly disease with 2 to 5 years expectancy once diagnosed, with no cure, and the drugs on the market bring only marginal benefits. So we use this bicistronic gene therapy platform to develop a first development candidate. We call it AVR001, which targets a genetically defined form of amyotrophic lateral sclerosis, which has mutations in the SOD1 gene. So we co-target with a microRNA SOD1 in neurons and astrocytes. So you go from a condition where basically you have loss of motor neurons and progressive paralysis to a condition where a single treatment with AVR001 can completely restore muscle function and muscle coordination. To convince you, I'm going to show you some rodent data. We have also tested AVR001 now in primates. You can see on the left transgenic mouse model of ALS where the motor function is compromised and the coordination is compromised in the swimming performance test, while a single intrathecal administration of AVR001 on the right can restore muscle function and coordination to wild type level. Now, our value proposition is based on 2 axes, this bicistronic gene therapy platform. We have also a discovery engine that allows to identify extremely innovative targets. So with this, we have in our pipeline a development candidate, AVR001, which has been tested, as I mentioned, in primates and rodents. We have 2 novel targets for ALS and frontotemporal dementia, but we are generally interested in neuromuscular indications with a novel target here. And with this, I see my time is over. So we need 50 million to bring AVR001 to proof of concept in patients and develop the pipeline. And I thank you for your attention, and my apologies to be over time.
It's okay, we didn't cut you off, you're doing fine.
Thank you.
One question is, how specific is the AAV, or are the AAVs that you're using? Are there many or any off-target effects?
So AAV9 is the serotype we use. It's part of a commercial product called Zolgensma, and it has a tropism for CNS cells. What confers the cell type selectivity is the engineering of promoters within the bicistronic vectors. So the AAV9 will transduce all cell types within the CNS, but the transgene will be expressed only in motor neurons and astrocytes. Important thing is that many gene therapies, even when administered intrathecally, will lead to leakage to the liver with high expression of the transgene in the liver, which leads to toxic effects. Now, what we've demonstrated in primates is that despite the fact that you have some virus in the liver, there's no expression of the transgene.
And you also showed a single dose in the rodent studies. Is that expected to be the same for humans?
Yes, gene therapies are generally designed as a one-off solution. So you should conceive these more as an intervention rather than a chronic treatment like the small molecule pill that you have to swallow every day, or the injection that you have to do every 3-4 weeks. That's the advantage of gene therapy.
Great, thank you so much.
Thank you.
So next to the stage, we'll be welcoming Alex Schuler. Alex is the head of Solvi, which is bringing about a new treatment modality using mitochondria, the powerhouse of the cell as we all know them. Alex, are you here?
Perfect, I am here. Thank you very much for having us, and thank you to the audience for taking the time to learn about what we all are doing. We are, as suggested, leveraging the therapeutic potential of mitochondria, so the powerhouses of the cell. Before I elaborate, I'd like to introduce you to Avery. Avery was born with a complex heart defect. In the procedure to save her life, she sustained an ischemic perfusion injury, leading to a cardiogenic shock. No treatment options, very slim chances of survival. No slides. That doesn't help, does it? I don't know why. Can you see them now? You should be.
They're coming up now.
Okay. Good to go. Thank you very much for letting me know. So Avery was without many options. But fortunately, she could be enrolled in a clinical trial at Boston Children's Hospital, receiving mitochondrial transplantation as a means to ameliorate the ischemia reperfusion damage. Within 24 hours, her heart came back. So today, she's a happy and healthy 3-year-old. Or to quote her mother, she's a rock star. This story inspired us to start Selvi to make this therapy broadly available and bring about mitochondria as a new treatment modality, starting in ischemia reperfusion, which arises whenever the flow of blood is interrupted and subsequently reintroduced. Medical conditions include strokes, heart attacks, organ transplantation, conditions we all know too well. Ischemia reperfusion injury is the world's #1 killer. What few people know is that at the core of the injury lies a mitochondrial dysfunction that arises during the ischemia, during that lack of blood flow. Without curing that deficiency, we cannot address the ischemia reperfusion injury. But what if we could replace and augment the mitochondria that are damaged during a heart attack? That's exactly what we are proposing and have shown to be highly efficacious. We're transplanting healthy, viable mitochondria into cells where these mitochondria have been distressed due to ischemia. This next generation cellular therapy has been developed at Harvard Medical School. I joined this group of world-renowned experts as a repeat entrepreneur in the medical space to take the technology to market. To put some numbers to the efficacy, in models mimicking a heart attack in a pig, we've shown that we can reduce damage to the heart by 60%. More than 20 preclinical studies were completed to show efficacy and safety, so enabling us to start the study that saved Avery's life. In that study, patients like Avery on ECMO are receiving mitochondrial transplantation to reinvigorate that heart muscle. And we've seen 80% myocardial recovery, where normally 29% of those patients only are able to separate from ECMO. This is huge. We've built a team of experts in the scientific, clinical, as well as regulatory and IP domain to take this technology to the next level and to the market. We've raised 5 million in our seed round last year in order to focus on 3 key milestones: productization, specifically off-the-shelf mitochondria, further clinical validation of our technology, and very importantly, showing that it's a real platform. Here, we're focusing specifically on aging and cell therapies, showing that mitochondria can have a benefit beyond ischemia reperfusion injury. So join us in turning patients into rock stars. Thank you.
Thank you, Alex. I think the first question is, can you walk us through any of the safety concerns that you're aware of here and how you're mitigating them?
Yeah, so since we want to go for an off-the-shelf product, clearly it cannot be autologous, but it needs to be allogeneic. So we need to have patient foreign injections. And so that brings about immune response risk that we have been addressing in a variety of immune response studies. We also are addressing the question of the genetic material that's inherent within the mitochondria and how that could affect the patient receiving the mitochondrial transplantation. And then there is, general safety of, not just immunogenicity, but also inflammatory responses and other potential effects of mitochondrial transplantation that we're looking into.
Fantastic. And have there been any other attempts at using whole mitochondria for therapeutic purposes?
Yeah. If there hadn't been, I would honestly be concerned. One of the companies that has shown a lot of traction has been Nmda Therapeutics out of Israel. They just actually struck a deal with Astellas for 20 million upfront. Pretty impressive. They're using mitochondria to enable their cellular therapy for mitochondrial myopathies, showing very nicely how the mitochondria are potentially a treatment modality that we have so far been neglecting, or rather not knowing enough about. So I think I'm really happy where we are right now. I think it's right time, right space.
Fantastic. It's finally nice to see the powerhouse of the cell be promoted to something a little bit more interesting. So I wish you the best of luck with that. And thank you for sharing your progress with us.
Thank you very much. All the best.
Great. So next we're welcoming to the stage Awa. She is with Nemozia, which is a company that's looking to raise public awareness of neurological disorders by finding methodologies to do early and regular brain checkups. Awa, are you here and able to hear us? You are on mute if you didn't know.
Hello, everyone. Are you able to hear me?
Hear and see you.
Okay. Let me share my screen. Let me know as soon as you have it. It works? Good. So hello, everyone. Thank you for having me. I am CEO and co-founder of Nemozia. At Nemozia, we develop first-in-class medical imaging radio tracer for early detection of neurodegenerative disease. Basically, our vision is really to have a brain scan as part of regular health check routine. And we think that in the next 10 to 15 years, imaging tests will be not only more accessible because of instrument being developed, which will be more scalable, but also more effective drugs and therapy will be developed by healthcare companies. Every hour of every day, our brain are controlling everything that we do, from our mood to the way we move, how we sleep, and how we interact. Yet today, neurodegenerative disease are diagnosed only after symptom appearance, which is already at a very late stage of the disease. There is no cure to reverse the degeneration. However, the best chance we have to stop at least the symptoms, to intervene much earlier when this disease first start to take hold. In fact, early detection will benefit not only the patient, the caregivers, but also the national healthcare systems. And that's exactly where we want to make a difference. With our tracers, we are able to detect disorders at subclinical stage, which means several years before current methods. Our ETH team, after over 7 years of research, has been able to develop the first NMDA, the first successful Nmda Pet Tracer. Nmda is a brain receptor, which is believed not only to be an early biomarker, but also a set for disease progression, right? So, and PET scanners are similar to MRI, but when MRI stays at bone or muscular level, PET are able to capture change at a molecular level. Therefore, by visualizing our NMDA brain, and specifically the gluN2B subset of these NMDA receptors, we can detect signs of the disease onset prior any visible symptoms. Our tracers are so innovative that we have filed a patent, and it's currently being evaluated in preclinical clinical trials. So today, the first indication that we have for these tracers are multiple sclerosis, Alzheimer's, and Parkinson's disease. But in addition to our NMDA tracers, we also have in our pipeline a second tracer, which will be more targeting cannabinoid for cancer and ALS. Let me walk you through quickly our high-level approach. As of today, we have already completed animal studies. We have done our POC on rodent, primates, and also have done first in man last year. And we also did some tests with post-mortem human tissue. In parallel, we have completed our term sheet negotiation with ETH for the exclusive license right. And we have also completed FTO search by Swiss IPI. We are planning to incorporate this January and having our pre-IND dossier preparation by end of Q1. We are looking to raise seed/Series A 12 million to cover for the first 2 phases of our clinical trials. So if there are any investors in the room, I would be really glad to continue the conversation with you. To go through this exciting journey, we have set up a very solid team with a mix of expertise. Dr. Ahmed and Dr. Aida are actually the 2 researchers who have started this project the last year under the supervision of Professor Amitame from ETH. And together they have won 2 SNF research grant and ETH silver medal. They are both not only authorized to manipulate radioactive products, but also hold lab management certification. Daniel, on the other hand, is our IT analytic guru, and he is currently completing his PhD at ETH. And lastly, on my side, I bring the business perspective with over 15 years in management consulting, particularly in the life science industry. Thank you very much for your attention, and I look forward to your questions.
Thank you. So you're addressing really 2 challenges. The first is the technical challenge of being able to put tracers into the brain and be able to see the progression of disease. And the second is human nature and the difficulty to get people into the testing facility early enough to be able to start an intervention. Can you talk a little bit about how you're thinking about changing the culture of early diagnostics for neurological conditions?
Yes, thank you very much. Actually, there are today more and more associations such as the Alzheimer Drugs Discovery Foundation that are doing a lot of campaign into raising awareness on mental health and early detection of neurodegeneration. And our side also, we have conducted several surveys to see how people will be reacting to taking early detection for Alzheimer. The main challenge that is blocking today is that they know that there is no cure. So the main challenge is why would we do this detection? But they are more and more motivated to see that basically if it is detected earlier, maybe there are better chance to stop the symptoms. And it's just now about really communicating and talking more about it.
And do you see a breakdown in those responses to the survey between those who have a familial history and just the general population?
Yes, definitely. So people who has already a family history are more inclined to take this early detection and to actually go and do it more in a regular base, the same way we do today for mammography and breast cancer. It doesn't have to be every year, but could be every 3 years or every 4 years, depending on the age of the person. And yes, but still, it's still a general concern, even for people who did not have any prehistory of the disease within their family. So we were very surprised actually to see that over 75, actually the exact number was 79% were willing to take the test, even if they don't have any family history.
I wonder, have you undergone a test yourself?
I would be the first one as soon as it's ready. I'm so curious to see our brain, and I think that's the way to go. We are focusing on any other critical organs in our body, but the main one controlling all over, we just wait to see what would possibly happen. I think it's a fascinating subject, and there are so much more that we can learn, and it's just the beginning of the adventure.
Fantastic. Thank you so much. And anyone in the audience who wants to be part of the adventure, please do let us know. Thank you.
Thank you.
Next, we're going to hear from Rajesh, who's with Nex Immune. Nex Immune is looking at a novel target for dealing with autoimmune disease and organ transplantation issues. So Rajesh, if you're here, we'd love to hear a bit about Coronin-1 dependent T cell activation. And we can see your screen, but we can't yet hear you.
Do you hear me now?
We do.
Okay, perfect then. So thanks a lot for the opportunity. Let me present Nex Immune. We are a preclinical proof of concept stage biotech at the forefront of T-cell based immunosuppression. We are a small team of 3 founders. Mr. Filippo Oliveri is the CEO. Myself, Rajesh Jayachandran, is a CSO. I'm a clinician by training and an immunologist. And we have Professor Jean Peters from University of Basel as our academic collaborator. In addition, we are backed up by a team of experts from medicinal chemistry, pharma, biotech, as well as finance. What we are trying to offer is a solution for the problem seen with non-selective immunosuppression. If you look at the drugs used for organ transplantation, like steroids, calcineurin blockers, and various antimetabolites, they non-selectively target the functioning capacity of every single immune cell subtype in our body. These cells are similar to the defense mechanism of your country. And what happens when the anti-defense mechanism is blocked? You get attacked. You get opportunistic infections, invasive cancers, and lifespan is a ticking time bomb. So how do we address these issues? It all started with a protein called coronin-1. We showed that coronin-1 deficiency by gene knockout, which is coronin-1 knockout, it selectively affects only a subset of your defense mechanism, which is T cells. All the other immune cell population, they remain unaffected and are fully functional. So we asked the question, what would happen in the context of organ transplantation, as well as in the context of autoimmune disorders? So in the context of organ transplantation, we perform both heart and skin transplant in both similar results. The wild type recipients, which are shown in red, they reject the new organ in less than 10 days time. Whereas in green are coronin-1 deficient recipients, which accepted beyond 100 days in the absence of any other immunosuppressive therapy. Likewise, in various autoimmune disorders, including multiple sclerosis, lupus, psoriasis, and IBD, it's only the wild-type animals, when triggered, they develop a very high disease score. Here you're looking at the example from multiple sclerosis, where the wild-type has a high disease score, whereas the coronin-1 deficient animals are strikingly protected. Interestingly, in terms of lifespan, both wild-type and knockout animals, they have a similar lifespan of 30 to 33 months, and during this time, they do not develop any opportunistic infection. When you challenge them with diverse virulent infection, they control it effectively, and they do not develop any cancers as well. So what we are suggesting is a shift from current non-selective immunosuppression to a futuristic selective immunosuppression, wherein by targeting coronin-1, you specifically block the T cell function. As a result, the remaining immune cells are fully functional. That prevents you from opportunistic infections, no cancers, and you can have a normal lifespan. So how do we do it in a clinical context? Towards this, we designed and developed an innovative therapeutic approach wherein a novel phenotypic screening assay was employed that targets coronin-1 pathway to deplete it. So we screened over 12,000 compounds, 12 hits were identified, and the top hit was named 5999. It also had good safety, and in a preclinical mouse model for psoriasis, it was able to strikingly suppress psoriatic disease development. You're looking at pictures of vehicle-treated animals, which have the plaque, scales, and erythema, whereas a normal pink supple skin with 5999. Our IP portfolio is rather strong. We have 2 published patents related to coronin-1 technology and a couple of additional patents that will include the novel screening assay and the screen identified compounds are in preparation with Woziers and partners. And now we are trying to raise $20 million that would enable us to go with the IND submission in 2024. A history of Nex Immune. We were just founded in May 2021. So far, we have successfully managed to raise $1 million. And now we look forward to raising a seed, 2 million, followed by a series A of 18 million that would enable us to go ahead with IND enabling studies and also go ahead with an innovative discovery program, which would enable portfolio diversification. IND submission towards the end of 2024 and a potential IPO in 2025. With this, I would like to thank you all for your attention, and I would like to inform that a recent publication in one of the top immunology journals, Immunity, was widely covered in various journals. I mean, here we were the featured authors in Immunity webpage, and this was also accompanied by a preview by Professor Ford from Emory Transplant Center titled Coronin-1, King of Alloimmunity. More recently, we also received a Swiss Transplantation Society Award, and I would also like to thank all the funding agencies that has enabled us to go ahead with the clinical development of this technology. I look forward to your questions.
Thank you, Rajesh. It's very interesting to see something so positive at the early stage. A couple of questions. So this is envisioned as a chronic treatment, or is it a single injection or a single application, and then it lasts for or persists for long enough to allow for good effects?
It will be chronic administration, repeated administration, what we anticipate so far. Yeah.
And so in between the administrations, how long do the effects last?
We have performed animal model studies, and we see that a single dose, the efficacy lasts for up to like 18 to 20 hours. So we are confident that it will go for a single day administration.
Okay. And are there effects to the immune system from having this chronic change to the T cells?
Except for a subset of T cells, which are the naive T cells, all the other immune cell population, it remains completely unaffected by this treatment, including the regulatory T cells. So it's very, very specific and selective for a subset of T cells.
Fantastic. And any other safety or tox concerns that you anticipate now?
So far, we have done chronic administration studies for a period of up to 14 days at a very high dose of 150 milligrams per kilogram body weight every day, twice daily. And we were not able to detect any side effects. The animals tolerated administration, they increased body weight. There was no changes in the liver function, renal function, serum electrolyte parameters. But I mean, still, we do not know what would happen in the context of like long-term, like 3 months, 6 months repeat dose toxicity. And those are the studies what we would like to do with the funding that we are going to raise.
Sounds like a good plan. Thank you so much for sharing with us, and we're excited to see how this progresses.
Thank you, Alicia.
Thank you. So last but not least, we're running a little bit ahead of schedule. So our last presenter is Anthony with PB&B. PB&B is also changing frameworks a little bit from what we've been speaking about so far, but they're trying to build or create a way to build fat for better plastic surgery results. So Anthony, are you able to hear and see us?
Yes, hello. I can just pull out the camera. Okay.
We'll just let that ambulance go by. Don't worry about that in the audience.
Okay. Hello, everybody. Anthony, I hope. Can you hear me okay?
We can hear you. Are you sharing slides? Because we don't see those.
Oh, yeah. Just 1 second. Okay, you can see the slides.
You're good. Yeah. And now I can't hear you. I don't know if anyone else is having the same issue.
Hello, can you hear me okay now?
Yes.
Yes, okay, excellent. So hello to Boston, Anthony Aho, here to tell you the story of PB&B. It's really the story of doctors coming together with bioengineers to render a lot of what is conducted in plastic surgery, both natural and non-surgical. So if you just remember one thing about what we do, we've created an injection which can increase the volume of fat. And well, why is that interesting? Well, you see, fat is the ideal material for a plastic surgeon to remodel the face and the body. So then the question was, what is the quickest pathway to market? And this is how we ended up focusing on facial aging. The root cause problem actually comes down to fat. As the face ages, fat tissue will lose volume, causing facial sagging and wrinkles. And it's typically treated with fillers like hyaluronic acid, but these give a synthetic result that lasts 6 to 12 months, depending on the product where you inject it. In our case, we're the first company that's created a technology that can specifically target the fat to revolumize it and in effect reverse this facial aging process. So we provide better natural results, and they actually last a bit longer than the competition. The product, we've made it as simple and efficient to use as possible for the doctor. Comes in the form of a vial with powder. He simply needs to add water, shake it, and then it's ready to be injected 30 seconds later directly into the fat tissue where they want to provide volume. How does it work? Well, the idea is actually quite a simple one. We want to increase the volume of fat cells. And to do that, we inject these microspheres, which act as a delivery vehicle for lipids, specifically oleic acid. This is the most prevalent type of fatty acid in your body. And once it's injected, it's releasing these lipids over the course of 2 months, superfeeding your fat. If you look at the tissue layer outside, well, the patient, they're getting their own tissue that's providing this natural long-term result that would last about 1.5 years. So just as, let's say, a wrap up, to date, we've raised 7 million. We're entering the clinical testing phase. We're doing this with, say, global plan for Europe, the US, and China. We have a patented technology. It's been approved in the US, China, and other major markets. We have demonstrated in our development the safety and superiority of our products and technology as compared to what exists in the market. And we've also scaled up and industrialized a lot of the aspects of the production. Right now, so we're actually raising 4 million followed by 25. This is to fund the full clinical testing phase, from a first pilot for the proof of concept, then the large scale pivotal trials as to gain market approval around 2025. Thank you very much, and I'd be very happy to take your questions.
Thank you. The question is there a single, is the single dose for each patient the same, or does the doctor have to decide to titrate up or down the amount given?
So that would really depend on the amount of volume to correct. At this stage right now, we don't know where the upper limit is of how much they can inject and how much of a volume they can correct just in one session. We do think if somebody just waits a lot of time, because, well, let's say this process starts at 35 years old. So, but if you wait until you're 55, you lost a lot more volume. So there we expect that you'd have to have at least 2 sessions.
So those would be 2 sessions within the 1.5 years that it persists?
Yeah, exactly.
Okay. And are there any concerns about the vehicle remaining in the skin?
So we're actually using a vehicle that's been used for over 25 years therapeutically. We're using PLGA. It's one of the most used delivery vehicles by the pharma industry. So actually, everything we're injecting, these are materials that have been used therapeutically for a very long time. We've combined it into something very new and a very different application. But generally speaking, all the safety data around what we're injecting, it's very well known and considered as very safe.
Fantastic. And then I know you've mentioned before that the face is not the only place where you could add these fats. What would be next after you do your proof of concept in face?
Yes, absolutely. So this actually works anywhere where you have fat. So it has a lot of different applications. For example, I think 10% of the US population suffers from loss of fat in the heel of their foot, especially in the older age. And that makes walking around pretty painful, and there's no actual therapeutic solution for this. So here, this would be a therapeutic application to help elderly in their mobility and be more comfortable. So that would be the therapeutic, and the second one would be for the body. There we would be actually pursuing the buttock augmentation indication because there's absolutely no product, and there's a big safety concern using surgery in that area.
Fantastic. So keep us posted on how it goes. I think all of us who are over 35 are very curious. Thank you.
Thank you very much.
Great. So that was our final presentation for the day. We've ended a little bit early, which I think is never a bad thing for busy people on the line. I do want to take a moment to thank everyone who joined us, both the presenters and our participants in the audience. And I also want to add that we at Swissnex continue to have other events that feature Swiss startups and sometimes even global startups. The first of which is next week on November 17th at 10 a.m. We'll be hearing from 2 panels that are focusing on challenges and opportunities in women's health, and also how to find that road to IPO as a femtech company. We'll also hear from 15 femtech entrepreneurs from around the globe who are sharing the way that they're changing the world of women's health. So for anyone who's interested in that opportunity, please go to swissnex.org and register for the event on our website. Thank you again for all of your time. A huge congratulations to all of our participants. We're looking forward to seeing the follow-up that comes from this presentation, and for all of you who indicated that you want to hear more from the presenters, we'll be getting in touch with you soon, and we'll also make the videos available. Thank you, everyone. Have a wonderful day. Enjoy whatever you're up to next and these 23 minutes that you get back as a gift. Goodbye.